this lesson is going to be about DNA. So you should have a copy of your notes sitting in front of you, and let's get started. The first thing, this is a picture in your notes at the top left, and then it says DNA means blank. This big long red word is what goes in the blank. Make sure you spell it correctly. When you're done, underline the D, underline the N, and underline the A. Now, this whole word is called deoxyribonucleic acid. We're already familiar with nucleic acids. That would be DNA and RNA. Deoxyribose is the name of the sugar found in DNA. D means without. The oxy refers to oxygen. And the ribo is actually the name of the sugar. So what this big long phrase, deoxyribonucleic acid, means is it is a nucleic acid that has the sugar ribose and it's missing an oxygen atom. So that's what the phrase deoxyribonucleic acid means. Now, below there's several statements. Fill in as many blanks as you can because most of this is a review. So fill in as many of those blanks as you can. Pause this to give yourself time. Okay, so this is what you should have filled in. DNA is found in the cytoplasm of prokaryotes. That would be bacterial cells because they have no nucleus. DNA is found in the nucleus of eukaryotes because we have a nucleus. DNA is a nucleic acid molecule. And each one of these colored words is what goes in the blanks if you don't have it already. DNA is arranged into pieces called chromosomes. And we've done this one already too. Chromosomes each have thousands and thousands of genes on them. So one chromosome may have 5,000 genes on it. One gene carries the directions to make one protein. And we know from the last test that proteins are made up of amino acids. So let me give you an example. Here's a chromosome. We know it's actually made up of 2 chromatids. And this is the chromatid here in a cartoon version. This is the real version. And this is made up of different genes. One gene on one chromosome codes for one protein, and in this case, we can look at hair color. Hair color is one gene, which makes the natural hair color of the person, whatever that color is supposed to be. Hair is made up of a protein called keratin. So one gene would be just for hair color. A different gene would be if you had curly, wavy, or straight hair. That would be a second gene. The third gene is how thick your hair is. Is it going to be really thick or is it going to be really, really fine? So each one of those is a gene. Okay, underneath your notes, called Cracking the Code, you have nucleotide is the simplest unit of nucleic acids. This is also a review. So the test you just took. And a nucleic acid or a nucleotide is the simplest unit. And here's actually a picture of that simple unit. You have a phosphate, a deoxyribose sugar, and a base. There's 3 things that make up a nucleotide, and here they are here. It's in your notes, made up of a phosphate, 1 deoxyribose sugar, and 1 nitrogen, which is given a capital N base. These are the 2 guys that discovered the structure of DNA, and they received the Nobel Peace Prize for doing so. And this guy right here, Watson, is actually still alive. He's 87 years old. Okay, so moving down, in your notes you have 4 types of nitrogen bases. You have adenine, which is abbreviated with the capital A, cytosine, which is abbreviated with a capital C, thymine, which is abbreviated capital T, and guanine, which is abbreviated capital G. Can you guess how they came up with the abbreviation system? If you thought it's the first letter of each word, then you would be correct. So these are the nitrogen bases, and here it is right here. This diagram here, this is the phosphate and the sugar. They're the same for every life form on Earth, whether you're a human, cockroach, bacteria, or whale. These 2 things are always the same. It's the base, the order of the bases. That's what gives us the code. And here's a diagram that's found in your note. You have the backbone is alternating, the circle is sugar, then here phosphate, this is the circle phosphate, deoxyribose sugar, phosphate, deoxyribose sugar, and so forth and so on. And these bases here are connected in the middle with these dotted lines, and those are called hydrogen bonds. Hydrogen bonds are the weakest of all chemical bonds. It's like walking down the hall with your significant other holding your pinky fingers as you're walking down the hall. It's going to be pretty easy for students to walk by and break your hands because holding pinky fingers, it's one of your weaker fingers, so you're not going to be able to keep that together as you're walking down the hallway. And that's very important for DNA. Now, okay, we are looking at, let's go back to this one, and the letters that bond together, it's already in your notes filled out. A and T always bond together. See, here's an A here with a T here. Here's a T over here with an A over there. And G and C always bond together. So here's a G bonded with a C, and this one is a C bonded with a G. They always bond together. So A and T's always bond together. G and C's always bond together. Now, towards the bottom of the front page, one word in nucleic acid language is 3 nucleotides long. So the number 3 goes in that first blank. And a word in nucleic acid language is called a codon. So in our English language, here's our little college dictionary. In our English language, words can be 1 letter long, like A, can be 2 letter long, at, 3 letters long, the word the, 4 letters long, the word band, and so forth. But in nucleic acid language, it is always 3 nucleotides long, never 2, never 4, only 3. And we'll talk later how the cell does that. Okay? So let's practice. Down at the very bottom of your notes, you have 2 things. This is the first one. If one side of DNA reads ATC, GGT, ATC, what will the other side read? Well, what letter bonds with A? Okay, if you thought T, you would be correct. So in the blank line, right underneath the A, you should draw a T because A and T always bond together. What's the second letter? Well, it's going to be an A. So this T up here will bond with this A here. What's this C going to bond with? G. These 2 G's are going to always bond with C. So C now, if you can't figure the rest of this line out, and there it is. So the T bonds with this A, the A bonds with that T, the T bonds with the A, and the C bonds with the G. And that is true for all life forms on Earth, period, no exceptions. All right, here's a second example, which is right underneath the first. If DNA reads CGC, AGC, ATT, There's 3 words or 3 codons here. What's going to go opposite the C? If you said G, you'd be right. What's going to go opposite this G? Well, that would be a C, because G and C always bond together. What goes opposite of this C is another G, because G and C always bond together. See if you can fill out the rest. Okay, so that would be the rest. So anytime you have a C in DNA, it will always bond with a G on its complementary or opposite side. If you have an A in DNA, it will always bond with a T. Now, when the nitrogen bases pair, it always works with one small base, always bonds with a large. So right here, here's a small base, T, binds with the large base, A. This small base, C, binds with this large base, G. It has to be that way. What would happen to the DNA ladder if 2 long bases joined together? Well, if we go back up to here, the bonds are this long. So between the nitrogen bases and the hydrogen bonds, it has to be this width. If you put 2 long bases together, then this piece here, the rung of this little ladder, is going to be too long and the DNA molecule breaks because it's too long. If you put 2 short bases together, then the rung doesn't go all the way across and the DNA molecule buckles or folds inward. So by actually having a small base and a large base always bonding together, DNA reduces the number of errors that can be made every time a cell replicates its DNA. So the pieces have to fit together, just like this picture suggests. Now, we're at the last piece, which is the top of the second page here. So DNA replication. Well, we already know to replicate means to copy, because that was in the mitosis chapter. So again, lots of repeat here, things you've already learned. The way DNA replication occurs, and this is your nice little picture. The DNA unwinds. Here it is. It unwinds. The white-sided DNA is your original strand. The black side of DNA is your new strand. So the old strand breaks in half, and then it unzips, kind of like a zipper, because DNA has to start at one end and it unzips all the way up to the other end. These hydrogen bonds that hold the bases, the actual nitrogen bases together, break. Okay, so example, these bonds right here break, exposing the A's, the T's, the G's, and the C's. When that happens, new nucleotides are added. And remember, a nucleotide is a phosphate, a deoxyribose sugar, and a nitrogen base. So this black strand are actually strings of nucleotides added on one after the other after the other. They're all added one at a time until DNA replication is finished. Do you guys remember which part of the cell cycle does DNA replication occur? Is it in interphase or is it in mitosis phase? If you thought interphase, you would be right. That concludes this part of the notes. And there it is.